Yeah, thanks for having me here. Been looking forward to this. Yeah, sharing kind of what is the vision for synthetic biology intersection with energy transition. I was also told to kind of keep this presentation more at the high level. However, I asked, our CTO, Tara, who is also my co-founder, and my sister to be on the call. So she's just joined us. So for the questions, if we wanted to get more technical, I have a bit of a safety net with Tara to answer those technical questions towards the end. Now, let me go ahead and share my screen. Okay. George, just give me the heads up if you guys see the full screen so I could take it
It
from
looks
here.
perfect. Looks great.
Okay, cool. Yeah, so we start with this slide because one thing that I like to mention is when we're all working towards this 2050 goal that we have for a sustainable future, net zero economy, the different ways that it's being characterized. But sometimes we think about the future as something that is kind of far into distant and is just kind of going to happen, whereas in reality we all have to work really hard to create the version of the future that we want to create. So when you walk into Cemvita's office, this is like large on the wall. The best way to predict the future is to create it. And for us as a company, we have this vision for reimagining and kind of repurposing heavy industries for the net zero economy, and a special focus on heavy industry because that's where a lot of the emissions are created. And as part of that, more specifically, we focus on the oil and gas and the mining industry, which I'll talk more about today. And yeah, just looking at the region and the states that are involved, there is a lot of legacy, oil and gas and mining activities, which I think, in one hand, present challenges when you think about energy transition, but as you see today, I think that also presents a lot of opportunities for new industries to be born out of energy transition and leveraging the legacy infrastructure within these estates. So the way that Cemvita approaches this is through a set of microbial solutions for sustainable extraction of natural resources, whether if it's hydrocarbons or if it's minerals for energy transition, metals, all natural resources. For sustainable production of chemicals and fuels through the use of microbes, or categorically calling it biomanufacturing. And then finally, with sustainable renewal of any waste that's created within that extraction and production process, and bring that back in a kind of a closed carbon loop. And the collection of these three is really how we envision a different way that oil and gas companies and mining companies could really reinvent themselves into 2050. And by the way, this is kind of at a higher level also, that really when you think about the relationship that we as humans have had with nature, I think that also needs to be adjusted. So a lot of this has been kind of extractive in one way, but now we want to kind of create a harmony with our relationship with nature. So on the oil and gas side and the mining side, and then also to end up producing the chemicals and fuels that we need to thrive as a species, but also take care of nature in a sustainable way, not just for us, but for next generations to come. So that's the nature inspired of this process in synthetic biology that we really love at Cemvita. So those themes basically feed then three verticals within our company as a platform synthetic biology company. The first one is carbon negative biomanufacturing. So this is the idea to use CO2 as a feedstock for biomanufacturing. There's several molecules that you could make from CO2. We'll touch more on that, but the flagship project that we have here is the bioethylene project with Oxy. Basically, the idea is there to use CO2 from a cogen power plant and turn that into bioethylene through engineered microbes that Cemvita is developing. The second category, the second vertical, is what we're calling here subsurface biomanufacturing. or essentially subsurface carbon utilization. This was born when we thought about, work right now sequestering a lot of CO2 in the subsurface, and there is a legacy around, utilization, which was mainly referred to CO2 EOR, or in-house oil recovery, the process of, using CO2 as a media to, pressurize the reservoir, change the miscibility of the oil to produce more of it. But if you take a step back and think about the subsurface or reservoir as a natural bioreactor, maybe we could actually do reactions in the subsurface. So that's how this vertical was born. And the flagship project that we have here is the Gold Hydrogen Program, which is the idea to make biohydrogen in situ in the reservoir as a new way of producing, zero carbon hydrogen. So we'll touch more on that during the presentation. And then finally, the third vertical is biomining. So this is for, turns out microbes could actually be turned into small mining machines for more sustainable extraction of metals. And that's across the mining workflow, from the initial bioconditioning, bio-cracking of rock, to biobeneficiation, to bioleaching, all the way to bioremediation. Microbes could play a role. We've already commercialized that when you think about wastewater treatment, but the same kind of workflow could be applied in the mining industry. So we're working on several projects there across two categories: one for base metals like copper, for electrification, and then battery metals with lithium, developing new methods for bioextraction of lithium. So when you look at this kind of landscape, really the idea was to have more of a holistic approach to energy transition. As I'm sure everyone is seeing now, the society is learning more about how these things are related. Initially it was just oil and gas companies really taking a beating, but now we're realizing, okay, well, that's one part of the puzzle, but then what about the metals that we need for EVs, and copper for electrification and different sources of energy? And within this, three verticals, we wanted to have more of a holistic approach. But you could see here how, when earlier I was talking about oil and gas and mining as the main focus areas, how they fit in. And with the oil and gas, it's both upstream, which is more for the subsurface biomanufacturing, and then also downstream for the carbon negative biomanufacturing. And there's a lot of ideas within the gray, kind of less explored crossover of these industries as well, which we could explore. So with that said, really I don't need to spend much time on this, but really the problem that we're solving is, on one hand, of course, yes, climate change. We need to help with kind of, on one hand, reducing the impact of our activities, on the other hand, reversing the damage that has been done, both in terms of the environmental impact footprint, also in terms of the amount of CO2 that's in the atmosphere, building into 2050. For our company, we have a very practical approach to this, and we want to work with companies, heavy industries categorically, that are, contributing 1/3 of global emissions, and helping them really reinvent themselves in ways that helps actually reduce scope 1 emissions, because that's the most difficult one. There is no shortcut around it. You can just buy a different type of electricity. And for scope 3, it's really hard for companies to control what the end consumer is going to do, and take on the responsibility for the emissions. But scope 1 is at the core of what they do. And because of that, it's really hard, because they actually have to change how they do things. And synthetic biology here offers an alternative, because these reactions, for the most part, happen under ambient pressure and temperature. So you have less, energy input into these reactions. And then on top of that, if you could use CO2 as a feedstock, now you could even get carbon negative and helping kind of offset emissions in other areas. I think that's the type of offset that I'll really get behind, as opposed to, planting trees somewhere else and continue on status quo operations, as a lot of these companies have been doing. And all of that is going to now be under, I would say, kind of a microscope with this announcement from SEC yesterday when it actually takes effect. So you're going to see a lot of changes in that space in the next few years. We have an internal goal at Cemvita to utilize 1 gigaton of CO2 by 2050. And, it's a big number, but it's actually quite achievable when we look at the pipeline of projects that we're going to commercialize by 2050. So we'll talk more about that too. Now, before going further, I just wanted to spend a quick minute talking about synthetic biology. For those of you who are already familiar with this space, and I know there's some on the call, please, I hope that you could just bear with me as I just kind of paint the lay of the land. What are we talking about? So synthetic biology is basically, giving us this ability to engineer microbes for new capabilities. We all know about microbes. They're living things. They eat something. They produce something. We know about breweries. We know about how yeast and E. coli could eat sugar, maybe make alcohol. But with synthetic biology, now we could actually redefine what we want the input to be. For example, maybe CO2 could be an input, and then what we want the output to be. Maybe instead of alcohol, we want ethylene, or some other organic acid. Those are possible now because of the cost drop in DNA sequencing and general tools around genetic engineering, CRISPR, and synthetic biology. So that's kind of the category and the engine behind what Cemvita does. And synthetic biology is by itself a growing industry really rapidly, 22% CAGR, which is something that mostly investors care about, but nonetheless, it's impacting different industries. And some examples of that to just look at the landscape of where it is started and where it is now, is if you think about not too long ago, like in 1920, we're using 10,000 pounds of pig and dog pancreas to harvest 1 pound of insulin. Until someone says, wait a minute, there's got to be an enzyme that is doing that, and it's a natural system. So can we go and figure out what is the genetic code for that enzyme and put it inside E. coli or microbe? So that's exactly what they did. And now, we produce insulin in sophisticated labs inside bioreactors. So it's gone from something that was very extractive, very kind of had to have the natural system, which we didn't have control over, to something that could be engineered, could be scaled, could be repeated, is independent of the environment, and doesn't harm animals in the process either. And so if you think about pharmaceutical industry, that's how most of basically the end drugs products are made today, through biochemical reactions. And fast track to what's happening now, that has come to other industries. And the last one is nutrients. And if those of you have tried the plant-based meats, either from Beyond Meat or Impossible or any of the other ones, it's really amazing because you bite into it and it tricks your brain. You really can't tell if it's meat or not. And there is a reason for that, is because they've taken the gene for heme or hemoglobin, which is the red thing that makes meat taste and smell and look like meat, from soy plant, and engineered it into a yeast, put it in a bioreactor, which now produces the liquid, and then you mix that with a bunch of plant proteins, and you have an end product that looks just like ground meat. So what has enabled that is the cost drop in the tools for synthetic biology. And what Cemvita wants to do is, we started with asking the question, is this the right time now to bring that into other applications, more, heavy industry applications, especially with the inherent benefits to energy transition? So that's what we've done. Now, to start off, the first application, the carbon negative biomanufacturing, really what we're doing is turning, these microbes that we engineer into tiny biomanufacturing machines or chemical plants, and defining what is the input, what is the output in the process. I'll mention that ethylene project just to give you a bit more details about that. When Oxy gave us that challenge, can you make bioethylene from CO2? Tara, who's on the call, actually looked at gene databases. So where is it that ethylene is produced naturally? and she came across, bananas, and most other fruits do the same too. As in the ripening process, they produce ethylene. So for me, I would think, well, that's cool. But for her, she's already thinking about the gene that's behind the enzyme that is actually doing that function. And at the time, this was 2018. In 2018, we already know the genome of banana. We could go find the line of ATCG DNA code that is responsible for the enzyme. We could take that and engineer it into a microbe that is using CO2 as a feedstock. And with further optimization, you could actually have a microbe that now makes bioethylene from CO2. So that's what we've been doing. We've completed the phase 1 of this project, which was, in a test tube scale, then going it to kind of 1 liter, doing further testing, measuring how much ethylene are we making, how much of the CO2 is being utilized, what's the energy input, all of that, building techno-economic life cycle assessment. And then since then, we've moved to phase 2, which is scaling the process up to 1,000 liters, updating the techno-economic assessment, life cycle assessment, gathering more data into more of a tangible framework that we could put in front of a chemical company like Oxy without prior fermentation or bioengineering background, but because the output is an Aspen model, process engineering is kind of a common language between us and them. And really that's what we're trying to do, is to enable chemical companies to also have biomanufacturing in their toolbox for their reactions. So the second one quickly is, subsurface biomanufacturing. And the idea here was to turn the reservoir, the subsurface, into a natural bioreactor. So gold hydrogen, we define that because, of course, if you have a new way of making hydrogen, you also have to pick a color next to, all these other colors that we have. And we pick gold actually because to pay a little bit of a homage to, how oil used to be the black gold, now hydrogen could be the new way that is going to change basically the business model for the oil and gas industry. As you look about, I was reading actually a Nature paper that came out a month ago that looked at, if we were to stay within the 2-degree temperature cap by 2050, the Paris Accord goals, 60% of oil and gas will have to be left on the ground, so it will be unextractable. And so for me, I think about it and it's like, okay, what if we think of that oil not as fossil fuels that we're going to produce and sell for fuels and feedstocks, creating more emissions, but what if we think of it as a natural feedstock that's in the subsurface? And if we already know the microbes that are in the subsurface that could drive certain reactions, what if we could optimize those by giving them the right nutrient to drive reactions of interest for us? And that's exactly what we did and came up with this pathway for fermentation of hydrocarbons and production of biohydrogen. And so the main reason that this is now getting a lot of traction is because the cost estimation is actually less than $1 per kg. Because if you think about it, there is very minimal capex that is involved, because we start with reservoirs that are already kind of in a petroleum engineering language, post-secondary or tertiary recovery, meaning we've already explored the natural pressure of the reservoir. We've already used water flooding, other chemical flooding methods, and there's still 10, 20 or more % of the original oil in place that we haven't recovered. So we could use these microbes in the subsurface to mobilize that and utilize that for this reaction. And it makes a lot of sense, in particular, to couple with blue hydrogen projects, if you're familiar, where we are basically doing steam methane reforming, but then in the back end, we're adding carbon capture and storage. So for those projects, because they're capturing the CO2, doing the storage, they have already identified the reservoir to do that. So we could have studied those same reservoirs, maybe a different pocket of those for production of gold hydrogen. So you think about conventional reservoirs in New Mexico, in Colorado, in Wyoming. There is a lot of these that is being explored kind of from early 1900s. And now they're either kind of already plugged and abandoned, or they're in the process of doing so. And some of them are actually orphan wells that could even be, emitting methane. And no one is kind of really responsible for that. And that's why they call them orphan wells. So there's a lot of opportunities that we could go back and turn them into subsurface hydrogen production facilities. So finally, just touching on this, the biomining application, I think I'll just cover bioleaching, because this was where we started as more of a low-hanging fruit because it was more obvious. With copper being such an important metal for electrification, but then to our surprise, we learned that 20% of the world's copper today is already bioleached. So traditionally, you go mine the copper and remove the ore, with open top mining, and then you create, when you stack them up, what they call heap leach, or like the heap. And from the top, they spray chemicals and mostly acids to leach out the metal, which then, from the bottom will collect for further processing and purification. So it kind of looks, the setup is kind of like a landfill. And you could do the same, but instead of spraying acids, you could actually spray microbes. And as those microbes make their way down, they do reaction with the ore in, capturing the metals in the process. So that could be more efficient by itself in terms of increasing the recovery, but also it would reduce the environmental impact of the process. For example, if you use sulfuric acid for leaching of copper, all that sulfuric acid has to go somewhere at the end. So most of the time it goes to a pond and will create what is called acid mine drainage, where you have, a pond with 2, 3 or more million gallons of pH 2 or 3 water, which a lot of times happens also to be next to a town, creating kind of risk to water sources for the town. And all the mining companies have this kind of either legacy assets or closed mine sites that they have to really take care of. And what they do is they have either access to or their own lime mines, where they bring the lime and kind of add it to the water to neutralize the acidity. But with a method like bioleaching, you would, eliminate the need to be doing a lot of that. And the overall vision that we have for Cemvita, by 2050 is actually to go even beyond that into in situ bioleaching, to where you could look at a place where it used to be a mine, but not be able to tell that it was, because these deposits, as they get deeper, open hole mining may not even be possible to do. And so bioleaching, is being done in the mining industry, but the part that was a bit of a surprise to us that has not been done is further optimizing those microbes for better recovery. So what they have is what, I've heard this kind of the term that is used as like bucket macrobiology, where there's been a species that they've isolated, 15, 20 years ago, and that's what they spray on top of the heap leach. Whereas we could really optimize those microbes that we isolate from the environment itself. Most of the time, they're natural microbes. We just optimize them, even sometimes without having to do kind of aggressive genetic engineering, and then deploy them back in the field for better extraction of the metals. So bioleaching and copper is a big focus area for us. We're working with several mining companies in doing that, but there is a bigger plan for biomining that we have. And also, interestingly, actually, our biomining division is based in Denver. We have our lab and office in Westminster by Denver, where we do the bioleaching test. So for those of you, if you're there, you're welcome to visit us. And then the rest of what we do in terms of biomanufacturing and subsurface carbon utilization is based in Houston. So the main question that you may have is like, well, okay, but how do we scale this up? Because every time you're introducing kind of a new framework, the scale up becomes the main challenge. And in this case, definitely, scale up is the core of what needs to be done. And what we've done is we've come up with more of a seamless process and really thinking about this in terms of taking risk off the table as we go. And so what we do is we define, these milestones in phase one. The goal is to just, okay, let's do this in the lab, show what's possible, measure what we can, couple that with whatever that's available in the literature to have a first one at a techno-economic and life cycle assessment. Techno-economic would tell us, is this economical, of course, but then life cycle is also very important because it would tell us when you consider altogether the energy input and output to the system and the sources of the energy, are you going to end up adding, CO2 to the environment, or taking some off, or you're neutral? And that's important, especially because some of these projects apply for, in the US, we have 45Q, which I'm sure you guys have heard about, $35 per each ton of CO2 utilized, which is most of what we do, and then $50 for each ton of CO2 sequestered. And there's movement around increasing those limits as well to $85 and maybe even more. Once we have that, we start to get a sense for, okay, is this possible or not? If the numbers don't look good, we would have kind of a few revisions to get them there. And if they don't, we'll just drop the project. Now, for those that do show us the good indication of economical and lifecycle viability, then we go to phase 2, which is a scale up to 100 times or 1,000 times, depending on what the process is. And the reason we're doing that is basically, if you think about what's happening inside the microbe, we need to boil that down to chemistry and then build it back up to chemical engineering, and then build it further back up to process engineering, and be able to have process flow diagrams and inputs to be able to start to build an Aspen model for what is this going to look like at the commercial scale. We do all of that as part of this phase 2, because for our company, we're not a university, we're not like a research institute. We have, our business model is licensing of these microbes to the end customer, so they have to be commercial for us to make sense to continue to work on those projects. So phase 2 would really start to make that tangible, and if we see the right indications there, and only then is when we go to phase 3, which is pre-commercial demonstration plant, which is going to be, in the case of biomanufacturing, next to the emission source, using live flue gas for that. And we designed that to be about 1/10 of the size of what we anticipate the full commercial capacity to be. And then we do that for, duration of time needed to de-risk that module. And then from there, we add more modules to get to full scale commercial. So this workflow, as you see on this slide, is most of the time for biomanufacturing makes the most sense. For biomining and then for subsurface, it's a little bit different. There's core studies involved. We take samples from the oil field or from the mine, and then we do bioleaching tests. For subsurface, we do core tests using core samples that represent the reservoir. Once we learn from that, that's kind of the scale up. Then we go to pre-commercial, which is testing, say within a well or two, kind of a huff and puff approach, pumping the microbes in or nutrients in, and kind of see what we see on the other side. And then for biomining, also doing the test on location, but in a very limited scope on a bigger leaching column in the field. And then from there, we go, to full scale commercial. Of course, all of these, as you could imagine, would require a very multidisciplinary team, which is the most fun part of this job, is to have, process engineer next to genetic engineer, next to petroleum engineer, next to, mining process engineer, working together, coming up with these different ideas and how to approach the problem, and testing, different solutions in collaboration with our clients. So that's what we've been doing in the past 3 years, is to really build up the team that is capable of, bringing these technologies to life. Alongside of that, of course, it's, one of those things that we're not intending to become a mining company or a chemical company or oil and gas company. So the business model is to collaborate with the industry, make the technology available to them. So we have customers in that space, but we've also really been fortunate to leverage corporate venture capital arms of big companies that are looking for the same solutions, but they may not have, the synthetic biology expertise to do it on their own. So we act as an extension of their team. So that's how we built this relationship with Oxy, BHP, Sumitomo, Mitsubishi, and others in doing that. And where we are in our company, I think, is that inflection point of proving up in the early stage of these applications. And then within the next few years, it's really where the rubber meets the road, where we have to go do this scale up and one by one bring these technologies to life. So I'll stop here. I know this was kind of a quick summary of everything. Happy to answer any questions on my end, about generally everything that I said. And if there's more technical questions, also I will, I'll pass it on to Tara to discuss.
Thank you so much, Moji. I really appreciated that. I have a ton of questions myself. I'd like to encourage questions from the floor as well. Please use your raise the hand feature or post them in the chat so that we can sort of manage the flow here. And while folks are thinking about that, maybe I'll pose one for you and Tara. And by the way, Tara, thank you too for being here. I guess, so I'm going to go sort of a non-technical direction here. And part of what we're trying to understand within the iWest region is regionally specific opportunities. And so you started your talk by noting that the region has a lot of historical mining and oil and gas, and that those provide sort of a one metric that might make what you're looking at attractive for this region. You know, the region has a lot of wind, a lot of solar. It's low in water typically. It's got a workforce that some of which has aligned with the historical mining and oil and gas areas. It has a broader set of workforce though also that's available. I'm wondering if you could maybe comment a little bit on, from a regional perspective, are there other things that you see that are really positive aspects for considering building on bioenergy economy? And are there things that might be challenging from the regional standpoint for going down that route?
Sure. Yeah. So I think you guys have been studying this, so I would love to hear your feedback too. But what we have seen is a state could actually create a scenario to allow for certain technologies to thrive. And you see that, for example, take California with low carbon fuel standard, you know, so that drives innovation in that space because there's a way that they're incentivizing that particular technology. I think the part that is really exciting about, when I think about Colorado, New Mexico, with the kind of the Permian Basin, the Rockies, is there has been a lot of natural resources extraction, and a lot of those have come to kind of end of life. Some are ongoing when you think about unconventionals. So if you think about it, there was the early extraction, and then with the advent of hydraulic fracturing, that kind of gave it a new life. What I'm saying here is, like, we're not done. Like, there could be a whole new way to go and develop those assets and repurpose those assets, whether if it's for the use of pore space for CO2 storage, in cases for CO2 EOR, but something like gold hydrogen could be, a lot of these states are studying how they could be hubs for hydrogen, and being close to where the actual use case is for that, especially where the talent is already there. I mean, that's why our biomining division is based in Denver, not in Houston, is because of Colorado School of Mines generate a lot of the experts, biohydrometallurgists, and mineral experts are in and around Colorado. So to have that vicinity is really helpful. In terms of like you mentioned, water, if there's ways to have access to renewable source of electricity, if it's with wind or solar or even geothermal, those are huge advantages, because even, for our processes, you will still need energy, and we don't want to use, in some cases, that's why we do the life cycle assessment, right? Because you can't have a process that, because of your energy use, it will eat into all the savings that we had. So we always look for areas where they do have access to renewable source of energy. So I think those are all huge advantages. I would say the challenge is, it's like, who's driving that? Is it like a big company that is just kind of really focusing on their own assets and, driving that? Is it the state that has kind of put together a plan incentivizing companies or different asset owners to come together for something like this? It has to be more of a coherent and integrated top initiative to do something meaningful. Universities and everybody else. And so that's kind of what we look for, is to see, are there hubs that have already kind of connected some of those dots, and maybe sometimes they have the pieces, but the missing link is like, well, what tangible technology can be deployed to help us with this? So not knowing too much more, but would love to kind of see what your thoughts are. And for everything that we said today, I mean, or anybody else that you guys have brought on to speak, would love to explore and working more with these different ecosystems in deploying our technology, but to the benefit of the end, users within the states.
Thanks, Moji, and I'll definitely follow up with you and discuss a little bit more where there may be some touch points within the region based on what we've heard already. We've got a couple questions both in the chat and hands raised. I'll go first to Rajesh Pawar.
Hey Moji, hey Tara, this is Raj. Thanks for the webinar. Couple of questions. You mentioned right off the bat that your target is somewhere around 1 gigaton of CO2 by 2050. Can you give more in terms of how do you see reaching those because you are looking at multiple different, vertical pathways, so to speak. Where do you see, the scales of those pathways going to, and what combinations of those you see getting to that 1 gigaton goal that you have?
Sure. Yeah. So to give everyone kind of an example of what gives us the confidence to talk about that is, say, for only one bioethylene plant that we'll build with Oxy would utilize about 2 million tons of CO2 per year to produce 1 billion pounds of bioethylene per year. Now, that's just one. Oxy will build the first one, and then we'll start to license them around the world. And a ethylene plant with a 1 billion pounds per year capacity is actually pretty small. Typically, it's like 4 or 5. So when you start to add and compound those building into 2050, that adds up to a sizable amount of CO2 that's utilized. Secondly, I think where you're going to see a lot of large volume impact is going to be where you could do reactions in the subsurface. Because that's where right now we're storing millions of tons of CO2 in the subsurface. But for us, that's kind of a waste if we're just capturing the CO2, injecting it in the subsurface, rather than using the CO2 as a feedstock within the subsurface for conversion into other useful chemicals, whether if it's methane through underground biomethanation, whether if it's coupling it with a different reaction for gold hydrogen, that's where you could use, millions of tons of CO2, and you could actually go to all the assets that are currently storing CO2 in the subsurface or doing CO2 EOR and utilize that CO2. The other thing is, as I think the only reason that CO2 storage and sequestration exists today is because we, as like the industry or humans, we still don't know how to use it as a feedstock because it's a very stable and happy molecule, so it doesn't want to react. But I think, if you think about photosynthesis, it's been doing that for billions of years. There's electrochemical methods of doing that. There's chemical methods of doing that. And in the next 5 to 10 years, a lot of those technologies are going to become better and better. And eventually governments are not going to continue to support paying companies to store CO2, and as companies figure out how to actually make money from CO2, then why would they want to just store it, right? So we're counting on some of these overall market movements, but we're going after, pathways that use a lot of CO2 in the process and pathways that will need to build a lot of that end product. So in that first carbon negative biomanufacturing vertical, we have 3 pathways. The first one is the bioethylene that touches on, polymers and plastics. Then there is the sustainable aviation fuel, which of course there's going to be a huge demand for that. And then finally, the renewable natural gas. Pathways from CO2 to these 3, I think will add up to quite a sizable amount of, CO2 utilized. And, so that's kind of where that 1 gigaton of CO2 utilized by 2050 comes from.
George, can I ask one more question?
Make it short, please. We got a couple that I want to get in before the end from others on the line.
That's okay,
yeah.
then I'll just hold it.
Yeah, if we could. Yeah, thanks, Raj. I want to. I'll go to the ones in the chat here in a second, but I wanted to get Huyen Din on first, please. Huyen.
Hi, thank you for the presentation. It's very interesting. I wanted to ask you, do you consider the hydrogen carbon negative hydrogen, and how do you determine that? Is it just the hydrogen production or the whole system itself? And the second part is more about what is the value proposition of the gold hydrogen you're proposing here versus just the renewable approaches, water electrolysis as an example, to making renewable hydrogen?
Yeah, so for the first one, this would be actually just carbon neutral at best. It would not be carbon negative because we're not in this gold hydrogen, the way we're doing it, we're not using CO2 as a feedstock. We're just fermenting the hydrocarbons and either capturing and re-injecting the CO2 or using downhole membranes to just only allow for production of hydrogen. So it will be carbon neutral at best, not carbon negative. And then the other one, I think, when you think about green hydrogen, there's assumptions that we could make for, and I know, DOE has the hydrogen shot with like 10 years, can we get it to $1? And there is a lot of assumptions that will go into that. I think the main one is going to be cost of electricity, right? And cost of electricity, we could assume to be, I don't know, like 3 kilowatt, per hour or 2 or 1, but it will never be 0. So there's always a cost that goes into that kind of sets the limit for how much lower we could get the cost to be. And then on top of that, we have the electrolyzers and the scale up that needs to be done there. So while that is going on for us, we think about, if you think about, the oil and gas industry, a lot of these assets, they've already spent the capex to build this asset. So for them, actually, a lot of it is a liability. They have to spend more money to plug and abandon these reservoirs. But if you could turn them into hydrogen production facility with pretty much no capex, the only cost that you have is for nutrients for activating the reaction of interest. And when I think about that $1 per kg, which is our current estimation, and if you use the downhole membrane, you could shave off another 20 cents from that. And depending on the size of the reservoir, it could start off with, places where it's like a depleted reservoir, but say by 2050, it could be that, there is a fresh reservoir that they haven't even explored. So from the beginning, they could just drill to turn it into a hydrogen production facility. So in that scenario, you actually have a lot more feedstock in the first place, so you could bring the cost further down. It just gives, some of these companies a different way to reinvent the infrastructure that they have today into the future. So it's not for all companies. So the value prop is more obvious for, oil and gas companies who want to reduce their carbon footprint, or any asset owner. But then again, also for anyone that wants to have access to hydrogen, we're finding a lot of interest because they want to be involved. For example, one of the members of this JIP is Chart Industries. And for them, they don't make the hydrogen, but they store it, they transport it, and they want to be involved in the process. So it depends on who we're pitching to, but a different way to produce zero carbon hydrogen at a very low cost. That's the value prop.
Great, thanks. There are a couple of questions in the chat that are related, so I thought we could try to take these in tandem. And I'll just read and paraphrase the two so you can sort of get the context. The first one's from Babetta Moroni. Great talk, Moji. And Babs wants to know, has genetic engineering for environmentally applied microbes been an issue with partners or local communities? And then Bo Hoffman has a couple of questions, one of which relates to navigating the regulatory landscape in the context of this kind of technology, and the other one relates to mineral owners getting royalties from this kind of process. Is that the same? So there are a couple in terms of the acceptance piece, and then one in terms of the royalty piece.
Yeah, so if Tara is still on, maybe I'll pass on the first question to Tara. But the mineral owners, I think that's a huge opportunity, and we'll look forward to building that business model and evolve it into something that we could build these kind of relationships where basically it used to be, the reason that you get the royalty was because you had the oil in the reservoir, and that evolved into, well, you could get it because you're renting your pore space basically for storage of, say, CO2. But in this case, you're renting the pore space and the use of the feedstocks that you couldn't get out anyway, but you would still have royalty from the hydrogen production at the end from the developer. So that's definitely something that we've been thinking about. Let me just check if Tara is on. If not, I could try to address. Okay.
Yeah, Moji, I am online.
Go ahead, Tara.
Thank you. Yeah, thanks for the question. In terms of the regulation, yes, that's true. For the when we apply this, the microbes in the environment, there are limitations in terms of the genetic engineering. So for this type of application, we don't do the genetic modification. What we do, we take samples from the environment. We do the DNA sequencing to understand the microbiome. And then we look for genes that we want, and if we find that that genetic pool in the microbiome, then we do the bioprospecting. We isolate the right population, and then the next step is the bioaugmentation. In some locations, we find the right strain. In some locations, we don't. So based on that, we decide about the right location with the right microbiome and a combination of the bioprospecting and bioaugmentation.
I see. Thank you, Tara. So yeah, it sounds like you are trying to not apply genetic engineering.
Yeah, that's correct. There are a lot of limitations when we talk about the environmental application.
Okay.
Yes.
Okay. Yeah. Thank you.
Sure.
Great. Thanks for that. Looks like we have some time, and so I'd like to return back to Rajesh for his follow-up question from earlier. Raj, are you able to get back on?
Oh yeah, thanks. So this comes to the processes or the different vertical pillars that you have. You're looking at different ways of manufacturing. One on the let's call above surface manufacturing, engineered system, so to speak. You have a lot more control, and as both of us coming from subsurface background, you lose that control once you start looking at the subsurface, focused process engineering. Do you see that as a challenge where you're looking at gold, hydrogen, and also the methane related processes, which will affect the way your processes will evolve, the engineering will evolve, and subsequently scale will evolve? And if so, I'm sure you have thought about it. Can you share how you're looking at overcoming those challenges, especially on subsurface where you want to have better control on what's going on?
Yeah, so that's an excellent question. I think, though, if we take a step back, well, first of all, none of this stuff that we talked about here are like easy things, like that's why they don't exist to the full potential today. But if you take a step back, on one hand, a lot of these products are natural products in the first place. I mean, that's why we say natural gas, right? Because it's methanogens making that. And then the part that is giving us a bit more confidence is we're now storing, hydrogen in the subsurface in caverns and salt domes, and now people are looking into depleted reservoirs also just for storage of hydrogen. So those guys have faced a lot of the same challenges. If you'd have told me, a few years ago that there are people storing, ethylene in the subsurface, I'll have a lot of questions like, how is this even possible? How do you know, it's not going to react with other things? How do you know it's not going to leak? How do you know you're going to put, 1 pound in, you're going to get 1 pound out? But a lot of that, there's a lot of legacy in the industry for doing that in the subsurface. And practically what we're saying is like, well, can we go one step further? Can we actually try to do the reaction also in the subsurface? And There is also more kind of on the oil and gas side. There used to be this concept of microbially enhanced oil recovery, where we use the microbes for production of biosurfactants to help with the mobility of the oil. Now, that wasn't, I wouldn't say the best application for doing reactions in the subsurface, but because of it, we learn a lot in terms of what works, what doesn't work, what species, how much nutrient we need to inject, what inhibitors we need to inject to make sure that, you know, we are not feeding some of the bad microbes like SRBs producing more H2S. And so because of all of that, what we try to do is to, when we do this study in the lab, is to try to understand as much as possible what are the other reactions that could happen. We're actually also doing the biogeochemical modeling of the subsurface, couple that with, say, a petrol model or a CMG model, you know, for the reservoir. And that's what's the nice thing about going to these old assets, because, you know, oil and gas companies, they already have a lot of data about that. And then you do it in a small scale, kind of learn from that and scale from there. So we have a few safeguards to kind of build up into what's possible, what is not. But, you know, there's places that they're producing, you know, natural gas reservoirs that are producing, you know, 5 to 10% hydrogen. So that's like fascinating. It's like, wait a minute, like, where is that coming from, you know? And if it's a biological source, are we at a place, I mean, like, you know, industry or humans to understand what is driving that, and can we actually control that, you know? So that's what we want to explore. It doesn't mean, you know, we're going to be successful just because we're going to try, but we're certainly going to try.
Okay, we've got a few more questions that have come in the chat. We've got just a few more minutes left, and so I'm going to read the first one here, and if you could do an abbreviated answer to that, we might be able to get another one or two in. And so this first one comes from Keegan Jalbert, and first, inspirational talk, thank you. If using enzymes and biomimicry for biomanufacturing with a CO2 feedstock, can you prevent the reverse reaction instead of favoring movement towards CO2 without energy input?
If I understand the question is that do we need another source of energy to push the reaction using the enzymatic mechanisms? So if the question is about the source of energy, yes, we need a source of energy. And in some of our strains, which are photosynthetic, like cyanobacteria, the source of energy is solar energy, sunlight. And to increase the capacity of enzyme, we do the protein engineering. We do also metabolic engineering to do the flux balance analysis, so to push the nutrients and carbon dioxide toward the products. We push the pathway toward the products. That's the engineering area. And then also for some other application in subsurface, we use other strains that they don't need light, but they use, for instance, minerals as a source of energy. So there are two different mechanisms. Some of them are photosynthetic, some of them are non-photosynthetic, based on the metabolic pathways of the cells.
Great, thank you. And this one's from Raul Gonzalez. For subsurface hydrogen production, do you expect the need to inject other nutrients into the subsurface to allow efficient microbial activity? If so, how can losses be controlled? Sort of related to Raj's earlier question.
Yeah, so we need nutrients such as source of nitrogen, phosphate, but for the source of carbon, basically we use residual of oil as a source of carbon.
Yeah, and that's an interesting question about the losses. You know, we just have to model and you know, have a good sense for the reservoir, some sort of a containment where this is happening. And even if there are losses, at the end of the day, it's like, well, you know, the estimation that we have is for each barrel of oil, we could produce 3 kilograms of hydrogen. Now, that's with a lot of assumptions, with certain input of nutrients. And for some reservoirs that could change, we just have to do the testing.
Fantastic. Well, listen, I know there's another question in the chat. I'm just going to read it, and we don't really have time for you to go into it because it's a real meaty one, and it's from Babs. It's, what are some of the technical R&D challenges you wish you had a solution for today? So I'm going to seed that with you for thought for further follow-up. I want to thank both of you for taking the time to be here today. It was a really enjoyable topic to hear about. You did a great job, Moji, in terms of inspiring all of us to think of new solutions in the area, and I'd welcome you to reach out and learn a little bit more about things that are going on in the area. And I'm happy to talk with both of you to give you a better sense of what's happening in iWest. I have sort of one parting question, and that is, I know we have some folks on from some of the different communities in the area. And if there's interest in terms of follow-up questions or understanding how they might be able to look at partnering, including not with necessarily big companies, but even small companies, I assume that they can reach out to you by the email that you showed earlier in the talk. Is that the best way?
Yeah, just Moji, MOJI at cemvitafactory.com, Tara at cemvitafactory.com, or go to our website, add us on LinkedIn. We'd love to hear from you. This was, it's like better in person, but yeah, please reach out. And yeah, we'd love to, have this be a tangible ending to helping with the iVest program as well.
Thank you so much, both of you.
Thank you.
Thanks.
Thank you for the opportunity.
Thanks everybody for attending too. All right, bye everyone.
Bye.
Thank you, everyone. Bye.